In this video, we're going to continue to look at how triacylglycerols are digested. And so we're going to talk more about chylomicrons. And chylomicrons will deliver our triacylglycerols to muscles and our fat tissues and dietary cholesterol to our liver. The chylomicrons will attach to binding sites on the inner surface of our capillaries and skeletal muscle and adipose tissue. And once they're attached, their triacylglycerols are hydrolyzed by extracellular lipoprotein lipases. Tissues will then take up the triacylglycerols that were cut off in their monoacylglycerol form and fatty acids. As these fatty acids are being reduced from the chylomicrons, they will shrink and shrink until they're cholesterol-enriched chylomicron remnants. So to follow along, so here we have our chylomicrons, and now we have our cholesterol-rich chylomicron remnants. These remnants will dissociate from the endothelium and re-enter circulation to be taken up by the liver, as shown here, and as a fire truck goes by. So our VLDL are also degraded by lipoprotein lipase in the capillaries of adipose tissue and muscle. So LDL are big red circles right here. Once they are breaking apart, the fatty acids are taken up by our cells, and the glycerols will return back to the liver or kidney to be converted to DHAP, and DHAP, as we've seen, can be used in glycolysis, but it has many other purposes as well. So when the VLDL, or very low density lipoproteins, when their remnants are in circulation, they're either going to be intermediate density, then low density. Half of these VLDLs are taken up by the liver, and you can see the other half are going to these extrahepatic cells. Now, animal cells, all animal cells need to acquire cholesterol, so they either do that by making it or they take it from low density lipoproteins. And if they take it up by LDL, they do so via receptor mediated endocytosis. So remember, endocytosis is taking something into the cell. So outside of these cells, you have these Ldl receptors, and that's shown here in the little Y sticks. And these are transmembrane glycoproteins that will bind ApoB100. These receptors are in clathrin-coated pits. So we talked about clathrin in Biochemistry 1. That's where you're going to find your receptors. And you get this clathrin-coated vesicle. Once you're in this vesicle, you're going to shed your clathrin and you're going to fuse with endosomes. Endosomes have very low pHs. So what happens is that the LDL particle will dissociate from its receptors. The receptors will eventually go back to the surface, as shown here. ApoB100, remember, is a protein, so you're going to break that down into amino acids. The cholesterol is going to be hydrolyzed, and you're going to get cholesterol and fatty acids from that. Now, receptive mediated endocytosis is very general, and it's used for a lot of large molecules that want to be taken by the cell. You can't use diffusion, too big. Usually can't use a specific protein, again, too big. So you need receptors that will bind to your target, and you just kind of envelop it into the cell. And a lot of these receptors will recycle from the plasma membrane to endosomal compartments and back. For LDL, it takes about 10 minutes from the outside inside too, so you're going to see 10-minute recycling time. New receptors, they're going to be made on the ER, travel through the Golgi until they eventually get to the cell surface. And the concentrations of cholesterol will control the rate at which these receptors are made. Because if you have a lot of cholesterol, you don't need to take in more cholesterol. So the rate of LDL will decrease, or the LDL receptors will decrease. So the next component we're going to talk about is high density lipoproteins. They transport cholesterol from tissues to the liver. It's the opposite function of LDL, right? So our LDL is for transporting cholesterol from the liver to different cells. Here we're taking cholesterol back to the liver. And that's because liver is the only organ that can actually take care of excess cholesterol. So if you have too much cholesterol in your body, liver is the only thing that can break it down. So, yeah. So this looks like a repeat. Half of VLDL are taken up by the liver via Ldl receptor mediated cytosis. So we already talked about that in the previous slide. HDL will bind to a cell receptor, and that cell receptor is named SRB1. So that's how HDL is getting into the liver. And that's it for this video. I will see you in the next video.